Today, we are going to go over enzymes. Before we get into them, we need to understand some background information. Enzymes are a type of catalyst. A catalyst is a substance that speeds up a chemical reaction. Now, why is this important? This is important because some reactions in our body occur so slowly that it will be impossible for cells to utilize them efficiently. By the time the reaction is done, we no longer exist. Catalysts, especially enzymes, work to get past this. In biology, enzymes are proteins that catalyze reactions by lowering the activation energy, which is the energy required to start a chemical reaction. These enzymes are not consumed by the reaction itself. The reactant that enzymes catalyze is called a substrate, while the specific area that substrate binds to is called the active site. Here, in an induced fit, enzymes will change the shape of the active site to allow the substrate to bind better. Enzyme activity can be reduced by enzyme inhibitors. Specifically, competitive inhibitors reduce enzyme activity by blocking substrates from binding to the active site. Here, the substrate has to compete or fight with this inhibitor for that active site. Another type of inhibition is non-competitive inhibition. Here, the inhibitor binds to this other site called the allosteric site, and this process changes the shape of the active site, preventing the substrate here from binding. Whereas enzyme activity can be hindered, it can also be increased. Cofactors are non-protein molecules that assist enzyme function. Here, the blue clay represents an inorganic cofactor, while the green clay is a coenzyme, which is like a vitamin or an organic cofactor. An enzyme with a cofactor attached is called a holoenzyme. Allosteric regulation is when molecules bind through non-covalent interactions to an allosteric site, which changes the shape and function of the active site. This may result in inhibition or stimulation of the enzyme's activity. Allosteric enzymes have two binding sites, the active site and the allosteric site. Here, this inhibitor is going into the allosteric site, which causes the active site to close and the enzyme to become inactive. This is allosteric inhibition right here. Now you have an activator going into that allosteric site, and now the active site's opening and the enzyme is becoming active. This right here is allosteric activation. Now we have a multi-subunit enzyme, or an enzyme with multiple active sites. For now, these active sites are closed and the multi-subunit enzyme is inactive. But when you have this substrate go into one of those active sites and be catalyzed, this allows the other active sites to open and the enzyme is now active. This is called cooperativity. Feedback inhibition is when the end product of a metabolic pathway acts as an inhibitor to an early enzyme in the same pathway. For example, the product from enzyme 1 is an intermediate one. And the final product then comes from enzyme 2. This final product binds to the allosteric site on enzyme 1, making the active site inactive. This pathway will only continue once the final product is used up by the cell. Because enzymes are proteins, their 3D shape can be affected by different factors, such as temperature, pH, and chemicals. Each enzyme has its own specific conditions that allow it to function optimally. When it comes to temperature, the rate of enzyme activity increases up to a certain point due to the increase in collision among the molecules. However, after a certain point, the enzyme will denature. When it comes to pH, enzymes function best at a specific level. Being outside their normal pH range can cause hydrogen bonds and enzymes to break, changing their shape and thus function.